Okay, hello everybody. Welcome to our last, hopefully our last set of slides for this section, our penultimate section, and then we will get on to our last set of slides about how control and tropism works. So just to recap, also just to bear in mind that this set of slides is a little bit demacard. There's a lot of randomness because I accidentally paused. So you've got the first video, which is an hour, the second video, which is only half an hour, and then the third video, which is an hour, and then this is the fourth. So please don't get confused with that 30-minute video in between. So this is the fourth set of lectures for the section. And just to recap the last section, we discussed that that the root morphology is very important. We know that the environment can give way to the way the roots grow. We know that the genetics of the plant will give way to the way the roots will grow. And ultimately, we know that obviously, very logically speaking, the area where roots are is that area that's going to be depleted of nutrients first. So now this last little bit that we need to talk about is biological nitrogen fixation. Now remember, I said to you that plants cannot use atmospheric nitrogen. So this N2 over here is atmospheric nitrogen. The thing about atmospheric nitrogen is it's a very strong, that N2, so I mean there's two nitrogen molecules, and the fact nitrogen, atmospheric nitrogen, is that it's got a triple bond. And that triple bond is very difficult to break. It requires a lot of energy, and plants cannot simply absorb atmospheric nitrogen like it does with oxygen because they can't assimilate it. Remember, assimilate, fixation, all of that, the same word. The plants cannot do it by themselves. So they're really dependent on this idea of nitrogen fixation, where atmospheric nitrogen is broken up and assimilated into usable forms for the plant, such as nitrites, nitrates, and ammonia. And this nitrogen fixation can happen in two major ways. You can get natural nitrogen fixation, or you can get unnatural. The unnatural will be the Haber-Bosch process, where we get our fertilizers from. And the natural way can be atmospheric nitrogen fixation or biological nitrogen fixation. And we're going to concentrate on biological nitrogen fixation. And ultimately, remember, we looked at SPAC and we understood how SPAC forms a cycle, a biogeochemical cycle. We looked at the Calvin cycle being a cycle. You've heard of the water cycle. Nitrogen also has a cycle. And it's this cycle that shows how microorganisms will break down and how nitrogen gets fixated into usable forms for the plant. Now, because the plant, when I'm saying usable forms, the plant can only absorb nitrates, nitrites, and ammonia. Once it has those forms, it can assimilate it what it needs to. However, we do have bacteria. Now, please note, not just bacteria. It can be any microorganism. It can be fungi, mycorrhiza, rhizobia, anything. Bacteria and fungi that can convert atmospheric nitrogen into usable forms, nitrates, nitrites, but specifically ammonium. And these are these that live in the soil. They are found in the soil. They can be free living, so they can live on their own, and they can take up this nitrogen fix it and put it back into the soil, and the plants can absorb it that way, or they can be dependent on the plants. For example, now we have a symbiotic relationship happening where the organisms, the microorganisms, will create a symbiotic relationship with the plants, where the plants will give them carbohydrates, and the bacteria, the fungi, etc., the microorganism will provide it with ammonia, the plant with the ammonia that it needs. This is you should automatically be thinking legumes, legumes, legumes. If you don't know what a legume is, it's part of the Fabaceae family. It's all your peas, beans, acacias, anything with a pod, anything that has a pod in it, sweet peas, all of that, they're part of the Fabaceae family, and they really are almost the I want to say groundbreakers. This is that family that we found really does have very high symbiotic relationships with mycorrhiza, or with microorganisms, where nitrogen gets fixed, and they really broke the ground in our understanding of it. And for a very long time, it was thought that it was only this family that could have relationships with microorganisms, symbiotic relationships, but we've later found out now that indeed it seems to be a lot of plants are involved in a symbiotic relationship. So, jumped ahead here on the slide, but we have symbiotic relationships where the microorganism provides the host with fixed nitrogen, or nitrates, nitrites, ammonium, or and in return, the plant gives it carbohydrates. This symbiosis can form a special structures called nodules, and this is a bean plant. And bean, it will be part of the Fabaceae family. These are the roots, and these are the special nodules. So what essentially happens here is the bacteria. This is a bacteria in this case. So for legumes, we found the bacteria rhizobia is generally what forms the symbiotic relationship. These things are species specific, so a specific microorganism will work with a specific family or a specific species. It can be fungi, bacteria, or something else, but or any other microorganisms. But what we know of is bacteria and fungi, and specifically with legumes, we look at a bacteria. So if you see examples here, just be aware that just because it's a bacteria, we're probably talking about beans. But what happens is we get these special structures being formed called nodules. So what happens is the bacteria infects the root hairs. It sends a infection thread into the root hairs, into the cortex, into that meristematic region, and it initiates uncontrollable cell division. These cells aren't given a job, so they are undifferentiated. And think of really think of cancer and tumors. So tumors are just clumps of cells. They serve no purpose, but they're just this big lump of cells where the cancer sits in. And that's essentially what's happening here. We've got these nodules forming by rapid cell division. They form nodules, and the bacteria are going to fix nitrogen in those nodules. Remember, the symbiotic relationship between a specific bacteria and a specific plant is species specific. Just because we're saying bacteria does not exclude fungi. And also that just bear in mind that the infection process is different for different species. So not all species are going to form nodules, Not all fungi are going to form nodules. As you're going to find later on, it's very species specific. So bear that in mind as we're going through this section. So remember, we're talking about these specialized structures. They're called nodules. These guys over here. And this is where the plant is really going to fix this nitrogen. Now, like I said before, you don't have to have a nodule in order to have the symbiotic relationship. The fungi or the bacteria can colonize on tissue and anchor on it, which we will see. The interesting thing about it is, okay, there is a specific enzyme which is generally found in the bacteria or the fungi, and it is called nitrogenase. And nitrogenase's job is to break that triple bond in between the nitrogen, atmospheric nitrogen. Now, nitrogenase is very similar to Rubisco in terms of it's a bit slutty and not necessarily going to bind with nitrogen only. If it sees oxygen, it's going to bind with oxygen. Well, I'm starting to think oxygen is the family wrecker here, the home wrecker, but nitrogenase works very similar to Rubisco. It binds, it's an enzyme, it's a lock and key situation. It binds with nitrogen, atmospheric nitrogen, and it breaks those triple bonds. But if it sees oxygen, it's going to bond with oxygen, and what's going to happen? That nitrogenase enzyme is going to be deactivated, and it's going to be absolutely useless. So in these nodules, the plant has developed leg hemoglobin, and this leg hemoglobin is a protein that binds with oxygen. So it will come out, it will bind with oxygen, so that oxygen's bound and married, and this leg hemoglobin, oh my gosh, this leg hemoglobin, I can't say it. This leg hemoglobin is going to watch oxygen like a hook, and it's not going to let nitrogenase near its man. So nitrogenase is only able to bind with nitrogen in order to do nitrogen assimilation or nitrogen fixation. Same word. So very this special structures and nodules um, needs to have no oxygen. It needs to be there needs to be no oxygen in the nodule, and in order to do that, the plant releases this leg hemoglobin, which is a protein which binds with oxygen, so nitrogenase can be available to only bind with nitrogen. So what I need you to make sure that you know is that nitrogenase is an enzyme. Nitrogenase is similar to Rubisco. It can have an affinity with oxygen or nitrogen, but if it binds with oxygen, it becomes unusable. You need to know that is a symbiotic relationship that forms for nitrogen fixing organisms or nitrogen assimilating organisms. There's these special structures called nodules, and ultimately we get the fact that this is genetically initiated. It's genetic. Once the bacteria once the bacteria infect, form a nodule, a gene is activated where leg hemoglobin is released in order to bind all the oxygen within that nodule so that it can be free of oxygen so that the nitrogenase can work properly. Okay. I am going to because it's very difficult to understand, but what I'm going to do is I'm going to draw how nitrogenase breaks these triple bonds, how nitrogenase works, because the enzyme that works with nitrogenase is molybdenum. And remember, we spoke early in the beginning how molybdenum is a micronutrient. It's still essential. It's essential for this. So the nitrogenase comes with the bacteria. But if there's no molybdenum in the plant, this process will not work. So you need to, so I will draw it for you. I will take a picture and I'll upload it onto Moodle so that you can see how nitrogenase is catalyzed by molybdenum, and it breaks these triple bonds of atmospheric nitrogen to give the plant access to nitrites, nitrates, and ammonia. Okay, so we've just spoken briefly about biological nitrogen fixation with specific regards to a bacteria and the legumes, and how this bacteria and legumes form a symbiotic relationship. They form specialized structures, etc., etc. But that's not the only thing. Mycorrhiza is fungi, essentially. It's a fungus and it's a plant, and the term mycorrhiza refers to the role of the fungus in the plant's rhizosphere. So if it's a mycorrhiza, it means that there is a fungus and it's in the root's rhizosphere, and together, whether it be parasitic, neutral, or mutualistic, they form a type of symbiosis, and they will work together in order to make it work. Now, this idea, now remember we spoke about bacteria and nodules and legumes. It seems that fungus is more widespread than bacteria. Uh, your non-mycorrhizal plants, meaning these plants that don't form relationships with fungi, will be saline, waterlogged, or disturbed areas like mining soils or on the side of highways, etc., etc. And the reason for this is, if any of you have seen fungus, and I'm not talking about a mushroom, it's almost, I'm going to flip a slide ahead. This is a fungus over here. It almost looks like cotton candy. It's very stringy. It's very fluffy, almost very cotton candy like. And the minute it gets wet, it gets washed away. So if anything is waterlogged or saline, like in the mangroves, it's not going to work. There's not going to be fungi. Fungi can't get wet. And then obviously it's very easy to disturb these collies. So when there is a disturbance, they don't like to be there. They like to be left alone. So again, the same with the bacteria and the legumes. We have a situation where the mycorrhizal fungi is given carbohydrates, and in return, the mycorrhiza will fixate that atmospheric nitrogen. Now, please don't get confused with a lot of terms, but these fine little tubes over here, these fine little hairs, if you want to say that, they are called hyphae, and a massive hyphae is called mycelium. Okay, so don't get confused. So here you go, one hyphae, lots of mycelium. And don't quote me on the pronunciations. Just as long as you know how to spell it, it's fine. Okay, now, so we've got this idea. We understand that there's rhizobia, that's a bacteria forming the symbiotic relationship with legumes, and they have specialized nodules. We understand that mycorrhiza specifically refers to a fungus and the root, whether that association be parasitic, neutral, or mutualistic. But there are two main classes of mycorrhiza fungi, and that is ectotrophic or Here we go. Arbuscular mycorrhiza. From here on out, I'm going to call them ECM and AMF. ECM and AMF. And what that means is ectotrophic. ECM means that they are growing in a close web on the surface of the root, and if they go inside the root, they are in between the cells. They are not within the cells. They are ecto exits, ecto exits. They are on the outside of the roots, and if they grow into the roots, they are in between the cells. Whereas AMF, it penetrates the cortex cells and it forms it kind of forms in that cell, and it uses that cell as a host cell to do the fixation in, and that's going to be where the swapping of carbohydrates and fixation is going to occur. So ecto outside, uh, AMF, it actually penetrates the cortex cells. So here we go. If you have a look, we've discussed this is the fungi, and you can see this is a plant, this is a stem over here. You can see the roots. Can you see the white section around it? That white section is called a mantle, and it is a bunch of hypha branching filaments that is called mycelium. So this is called a mantle, this white, almost solid structure. And you can see here they did an experiment. This is with fungi, this is with mycorrhiza, and that is without. So they were planted the same time with that, and you can see the difference that nitrogen fixation has on the plants. And have a look at the soil. The soil looks similar to the introduction video where we were in the bluff and we saw the bluff soil. It looks exactly the same color, lacking in nutrients. So having the mycorrhiza is almost vital in helping with nutrient poor soil. So this is just a picture. You can see the cortex cells. You can see, remember this is ecto. They're not going to the cortex cells. They're going in between the cortex cells. They're forming a mantle. And this hartig net is actually just the in between the cells, forming like a net-like structure in between the cells. Okay, and you can see very clearly that the hyphae are much finer than the roots, and because they actually have reach beyond the nutrient depletion zone near the roots. So you can see the root stops there, and this mycorrhiza, these fungi are reaching further, and they're going to absorb the nutrients here, and they're going to send the nutrients through the mantle into the cortex cell. They're going to fix the nitrogen, and there's going to be an exchange here. So ECM, we've already discussed this. We know that it forms a mantle both outside the roots and within the air spaces. It goes around the cortex cells. It does not penetrate. There's no intracellular penetration. It does not go into the cortex cells, and as a result, that is called hartig net. And then you can see our AMF. Sorry, let's just go back a little bit. That hartig net is just a network of inward growing hypha that extends into the root, penetrating further into the root, into the cortex area, but not within the cells itself. Okay, so then we have this AMF, which they do not produce a mantle because they actually penetrate within the cortex cells, and they almost take over the cortex cells, and that's where the process is gonna occur. This is where those are reproductive. So this is fungi. This is the fungi's reproduction section. These are vesicles. These are going to be where the nitrogen gets fixed. And here we're going to have the exchanging of processes. Okay, so this AMF there can be 3 types, and these 3 types are literally just specific mycorrhiza that have specific relationships to specific families. So that's all you really have to know, is that they have so these are 3 types: BAM, Ericoid, and Orchidioceous, and that's just orchids, and these are Ericaceae. So this is just, these 3 types are just specific types, and vesicular is just vesicular will be those forming these specific vesicle regions. Okay, so what I need you to know is that there's 2 types of mycorrhiza, ECM and AMF. And in AMF, there's 3 types: VMA, Ericoid, and Orchidaceous. And the only thing you need to know about these 3 is that it refers to specific families, but I need you to really know that there are 2 types of mycorrhiza. And here you can see a lovely differentiation between the 2 of how ecto is really doesn't penetrate the cortex cells. We have a mantle, whereas here we have vesicles being formed in the actual cortex cells. Here's another diagram of it where you can see the differences, where it goes in between the cortex cells, whereas here it actually penetrates the cortex cells. Here's a picture of the vesicle form within, and those netting. Remember, this will be specific for AMF. And you can see here how it forms, how we get vesicles. You can see it. This is where where this is going to be stored. This is where they're going to transfer nutrients, etc., etc. Okay, so whether it's outside or inside, we can have fungal spores inside and outside the roots, which plays a very big role in the growth of the plants, etc. And just a side note, fungus within the roots, it seems to be the major problem with fungi or bacteria for tissue culture. So if you're going to take Paula Watt's module next year, tissue culture is very clean. It's with media. Google a little bit of it, and our major problem is this fungi that seems to have an affiliation with the roots. We are starting to discover that most angiosperms are associated with this mycorrhiza. Not only does it facilitate the uptake of trace elements and other elements like essential elements and help fixate nitrogen, it really does improve the absorption, all of that, and by extending past what the roots can reach. And plants really do better with this fungus than without it. Okay, so you've got this YouTube video which explains how mycorrhiza can be future for fertilizer. It's very interesting, and that is honestly the section. The section is done, it's complete, and I just want to quickly go up to the top again. Okay, so I need you guys to let me just get up to the top. Okay, so I need you to understand that soil is complex. Soil is generally negative. Uh, the clay particles, the quartz, they are generally negative. We have not only microcolonies of bacteria, but we have fungi as well. We know the difference between nutrient poor and nutrient rich. We understand the importance of fertilizers. We acknowledge that there are many different ways of classifying essential elements. We acknowledge 17 macro and micro elements, and we acknowledge them based on Aaron and Stout, 1939. We use their classification. There are other types of classification, such as the groups. However, there seems to be a little bit of disparity between the fact that they seem to do biochemical functions, but multiple nutrients can do the same function. We understand that in order for an element to be essential, it needs to be a part of the structural components, it needs to create energy, and it needs to activate or inhibit enzymatic activity. We then discussed how hydroponics can be used to establish the deficits or nutrient deficits of certain elements. So by manipulating the solution and removing one element, we could see how the plants, what happens to the plants when we remove a specific essential element, and how this element could not be replaced by another essential element. We had a look at this idea of transport, primary active transport and secondary active transport. We acknowledge that both of them need energy in order to work. We know that cation exchange capacity is very important, and it is the degree to which soils can absorb and exchange ions. And the higher the cation exchange capacity, the more minerals are within that soil. We know that soils prefer 5.5 to 6.5 pH, and we understand that as the plant absorbs these, it can either make it more acidic or more alkaline. We understand a dose response curve, and especially that we could have, in terms of essential elements, where we don't have enough and then we have excess, but in terms of it becoming toxic, we could eventually have death. We understand that just because you are the same species doesn't mean your roots are going to be identical. It depends on the environment in which you grow. We understand that just because you are different species and in the same environment doesn't mean your root morphology is going to be the same. We know our 3 different meristematic regions and the importance of that. We acknowledge where the cortex cells are. We acknowledge that the areas, the rhizosphere closest to the roots, are going to be depleted from nutrients and minerals first. And then we acknowledge this idea plants cannot use atmospheric nitrogen. They need something to fix it for them. So we have rhizobia that form a symbiotic relationship with legumes, and how we have nitrogenase Nitrogenase can only work in the absence of oxygen. We have special structures called nodules, and it is gene activated. We have fungus, which is mycorrhiza. So mycorrhiza really refers to the fungus and the root, that process. It can also form a symbiosis that could be parasitic, neutral, or mutualistic. We know that there's two major classes of mycorrhiza, ecto and AFM. We understand that AFM is internal, and we're going to start forming these vesicles, which will have spores inside and outside. We understand the terminology, the mantle, the hartechneck, etc., etc. And we understand that this fungus really does help plants grow, and without it, we definitely see a lack of yield. And that is all for plant mineral nutrition. I hope that you understand everything. Take it slow. And I will definitely see you guys on Discord, and I will see you guys in